really well on your tests. That's the easiest test of the semester, but I think you should still be very, very happy. You got your class average was above an 80. I think yours was an 84, which is amazing. Keep it up. And for those of you who didn't perform as well as you wanted to, I would love it if you would take a minute to come see me in my office. Because although I do not know how your individual brain works, I have seen a lot of successful students and a lot of unsuccessful students, and I can share with you what seems to work well for most people and what hardly ever works at all. Okay, and I'll let you troubleshoot your tests and all of that kind of stuff, and you can take as many notes as you want to. So I would encourage you to do that before the material gets away from you. Okay. Today, this is my favorite stuff to talk about. So I think where we left off was talking about ionotropic and metabotropic receptors. And wait, we talked about mechanisms of getting rid of neurotransmitters in the synapse already in here? Okay, so let me just give you a visual that I didn't give you guys last time. Okay, so before we move on, I mentioned the words ionotropic receptor and metabotropic receptor, but let me show you, okay? An ionotropic receptor is simply a receptor that the neurotransmitter binds and directly opens or closes an ion channel. And of course, that could cause depolarization or hyperpolarization of the cell. Does that make sense to you? And it's relatively quick compared to this one. This one is the one that is much slower to accomplish the same thing. Neurotransmitter binds. There are 3 other proteins that are stuck in the cell membrane. This one has to slide through the cell membrane and smash into an ion channel receptor, and that's how you open the ion channel. Slower or faster? Way slower. I can't believe that they evolved at all. But they did. There's 4 different proteins to accomplish what one could do. In Texas, we call that going around your ass to get to your elbow. You guys ever heard that expression? It's a good one. You should use it whenever possible. So, these are really, really common. They're metabotropic. They're slower, but they're going to accomplish really the same thing, honestly. Okay, and we'll talk about each of these again before we finish this set of notes. Okay, so the last time where we were was talking about ways of getting rid of a neurotransmitter once it's in the synapse, right? Okay. And can anybody explain to me why it was important to get rid of a neurotransmitter once it was in the synapse? Why you wouldn't want to generally just let it sit there after it's done its job? Toby?
You need to communicate that another one came. Communicate that another one came and has to be empty again.
Right, exactly. So if you just let it sit around, you would lose your frequency coding, right? You would lose your capacity for frequency coding. So if you let it just sit around every single time you released it, then you wouldn't be able to tell whether the presynaptic cell was releasing frequently or not releasing at all, right? That makes sense? Okay, so the mechanisms of getting rid of the neurotransmitter once it was in the synapse were 4. This one only shows 3 of them. Diffuse away, not going to be super fast. Reuptake it into the presynaptic cell, repackage it so you can use it again, right? The postsynaptic cell here has an enzyme present that has the capacity to change the shape of the neurotransmitter so that it will not continuously stimulate, okay? And we will see today 1 or 2 different examples of when that occurs. At an acetylcholine synapse, this one, what acetylcholine does at most of its synapses is there's an enzyme on the postsynaptic cell called acetylcholine esterase. And what it does is it takes acetylcholine and breaks it into choline and acetic acid. And choline is the more precious of the 2 so that it reuptakes the choline and then makes it back into acetylcholine. Does that make sense? But conceptually, does it make sense to you that if you change the shape of a neurotransmitter, it can't activate the receptor any longer, right? So that's all you're really doing is changing the shape of a neurotransmitter. Okay, and then there's another one that we'll see, monoamine oxidase. Monoamine oxidase is specifically for a different type of synapse. It's for dopamine, norepinephrine, and epinephrine synapses. And what there is on the postsynaptic cell, there's an enzyme that breaks those down so that they can't stimulate the postsynaptic cell again. That makes sense? Okay, so we'll see that either today or next time. And then the other the other way of getting rid of it was the astrocyte plays ball boy, right? Takes it, shoves it back, takes it, shoves it back. Okay, so now what we're going to get into next is how does a neuron communicate with the next neuron specifically? So one of the things that I've sort of implied to you that's a little bit of a fib is that the neuron to neuron pre to post relationship is 1-to-1. And it hardly ever is, especially in, like, think about the cerebral cortex. Do you think a cerebral cortex neuron has connection to one other neuron? No, it doesn't. So usually, especially in the brain, the relationship is more like 15,000 synapses per neuron. Why didn't I teach you 15,000 at a time? Because it's just one 15,000 times over. It works the same way, right? So what we're going to talk about is the relationship of pre to post is seldom 1-to-1. So what kinds of relationships exist? All different kinds, but let's simplify them to 2. First, there are 2 different sort of speaking to one another relationships between pre and post. This relationship is called convergence, in which you've got a whole bunch of presynaptic going onto a single post, converging, if you will, onto a single post. That make sense to you? Okay. So this one is called divergence, and divergence is when you have one presynaptic cell that splits and communicates for a whole bunch of posts. That makes sense? Convergence and divergence. So let's talk about this convergence business in just a second. Okay, so convergence has multiple presynaptic cells communicating to a single postsynaptic cell. And so what we see here is they're trying to represent to you that the green ones are excitatory synapses, and the red ones are inhibitory synapses. Go, stop. Yes? So what is this cell going to do? This cell is going to take in all the inputs, and it's going to sum them or integrate them. And what will really matter is whether the axon hillock gets to -55 or not. Does it matter how it got there? No, it doesn't matter how it got there. So what happens is you can have a bunch of different inputs on a single neuron, and some of them are saying stop and some of them are saying go. What wins? -55 wins, right? If you get there, you get an action potential, and if you don't, you don't. So why the hell would you be doing this? Like some yeses, some nos. Well, the best example I can come up with for you guys is trying to get your butt out of bed in the morning when you didn't get enough sleep, but you got some crap to do, right? So what is the alarm saying? It's saying get up, right? What is your body saying? It's saying sleep, right? But then what is going to be most insistent? Well, if the alarm keeps going over and over again, then you have a whole bunch of goes, right? You got a few stops. So which one wins? The ones that's most persistent or has the most inputs or lasts the longest is going to win, right? So this one is saying, I already paid for the damn class. This one's saying, I'm exhausted. The other one's going to say, you're going to have to take the damn class again. So whatever inputs are all summed, and the place that is important is the axon hillock. If the axon hillock gets to -55, you get an action potential, regardless of what the initial input was. Okay, so that is convergence. Divergence means that this one presynaptic cell would be controlling simultaneously, just in quotes because the lengths of the branches are a little bit different, right? But wouldn't these kind of work as a cohort, right? Okay, so maybe this one is a hypothalamus cell that is taking input about temperature, about emotion, about metabolism, about light, all taking that all information in to figure out whether to send an action potential, right? And this one's a hypothalamus cell that says, I want to send an action potential and I want to control cardiac muscle activity. I want to control other portions of the brain. I want to increase the metabolic rate of the thyroid. So it's sending it out to all of these places, right? Convergence, divergence. Got it? Okay, so basically what's happening is the axon hillock of the postsynaptic cell will integrate or sum all of the inhibitory and excitatory impulses. If you get a threshold, you get an action potential. If you don't get a threshold, you don't get an action potential. It's basically the all or none principle again, right? Okay, and of course, we talked last time about the fact that new synapses actually are forming during sleep. That's what the new research suggests. So let's talk about what I would mean about and what a new synapse would mean. So I want to put these two together right here, this pre, this post. okay? What if I have a repetitive activity or reinforcing a bit of knowledge that makes these two actually follow one another over and over? What can happen when that occurs over and over again, instead of 4 synapses, I can form 4 more. And can you see then that if this one actually forms 4 more synapses off of this branch, can you see that? That you would have a much faster response between those 2 cells with every firing, right? So I'm not saying you're forming new cells, but you are forming new synapses from existing cells. And this is called synaptic plasticity, and the more you use a behavior or the more you recall a bit of information, the more synapses you actually have the capacity to form. And most of that occurs during sleep. Good? Okay. So now let's talk about getting that postsynaptic cell going. Okay, so go back here for just a second and say, okay, so generally the relationship of the anatomical relationship between presynaptic and postsynaptic cell is not 1 pre to 1 post. So I already said that. And now I'm going to tell you that the physiological relationship is also not 1 pre action potential to 1 post-action potential. So just because I had an action potential and neurotransmitter release here does not mean I always get an action potential there. Okay. Generally what it is the presynaptic cell has to release neurotransmitters multiple times for the postsynaptic cell to get an action potential. Okay. So this part is going to be the slowest part of the nervous system. The synapse is going to be the slowest part of the nervous system. The action potential may go down an axon 250 miles an hour, but then it has to start all over again at every single synapse. And now I'm telling you that a single neurotransmitter release is generally not going to get you an action potential. So you're going to have to release it 4 or 5 times. Okay? So what happens here is you have what's called synaptic delay. The slowest part of the nervous system is the synapse. How slow, how much does it delay? It delays between 0.2 of a millisecond and 5 milliseconds, but you literally could not possibly notice that because your noticers work with that synaptic delay, right? There's no way in hell that you could ever notice, right, that synaptic delay. That is fastest in the nervous system, slowest in the peripheral nervous system. Fastest in the central nervous system, slowest in the peripheral nervous system. That make sense? Okay, so let's see how you get that synapse actually going. If it's not always 1 action potential here, it gets 1 action potential here. Here are 2 different ways of getting that postsynaptic cell going. So we are going to look at 2 different forms of summation. Okay, before we go any further, look up at this picture for me for just a second. Okay, and so at first, all I want you to do is pay attention to this presynaptic and this postsynaptic cell, just A, okay? A and the postsynaptic one. A is green's probably trying to represent what kind of synapse? Excitatory or inhibitory? Excitatory, okay, so just pay attention to A for just a second. We'll pick up B and C in just a second. So the first way that you can get the postsynaptic to an action potential is you can do something called temporal summation. And temporal implies what other than the bone and the muscle? Time, exactly. Okay, so with temporal summation, what we can do is if we stimulate A, yeah, and we get a little depolarizing graded potential, and then we stimulate A again after I've repolarized, then we get no summation, right? But what if I stimulate A instead of A, pause, A? What if I go, AA? Can you see that what I'm doing is I'm actually adding to the depolarization, right? I'm summing the depolarization. Now, the question that probably came up for at least one of you is, what about refractory period? Do I have a refractory period when I have graded potentials? No. Refractory periods start when you have action potentials. Yes? So there is no refractory period for a graded potential. So you can totally stimulate A again. Does that make sense? Okay. Now, I'm not saying that A, somebody asked this in the last class and it was a good question. Doesn't A here have to deal with its own refractory periods? Yeah, it does, but we're not faster than that, right? We're letting it release and then release again, okay? So what I'm really doing here is I'm adding to maybe the number of open sodium channels and getting a bigger depolarization, right? That makes sense? So if I stimulate A by itself, I just get a small depolarization, but if I stimulate A frequently, I get a bigger depolarization. Doesn't that really correlate to the frequency coding thing that we've been saying anyway? It totally does, right? Okay, so this is called temporal summation, okay? Temporal summation. We're getting the postsynaptic cell closer to or at an action potential by more frequent stimulation on the presynaptic cell. Yeah? Got it? Okay. And then, okay, let's look at another kind of summation. The other kind of summation is called spatial summation. You ready? Spatial summation is going to use A, B, and C in some combination. Okay, so please note that A and B are excitatory and C is inhibitory. You with me? Okay, so what happens if I stimulate A by itself, just a little depolarizing graded potential. Stimulate B by itself, same thing. Stimulate C by itself, hyperpolarizing graded potential. But what if I stimulate A and B simultaneously? Right? Does that make sense? So does the axon hillock care which one opened? No, it doesn't care, right? So if I stimulate A and B simultaneously from two different spaces or places, I get spatial summation, yeah? But can't you sum to 0 too? What if you stimulated A and C at the same time? Right, they just cancel each other out, right? And that would happen when you were like changing the activity of a neuron. That make sense? So spatial summation. Okay, question for you. Which one, temporal or spatial, requires convergence? Yes, spatial requires convergence. Does that make sense? Okay, now one last concept check question for you about this is, what is the fastest possible way that you can imagine to get this postsynaptic cell to threshold? Spatial summation. Is there any reason you couldn't do spatial summation very frequently?
You can do both at the same time.
Yeah, you can do them both at the same time. A and B very frequently. Yeah, you could do both of them. That make sense?
If you had like 10 of them all doing it at once, it wouldn't matter if they did it together, right?
That's true. If you had 10 of them all doing it once, but if you had this one, right, that was doing it 4, right, and then doing it really frequently. Yeah, go ahead.
So you were talking about, say, the one where your cursor had the four presynaptic clefts.
Oh, this one? Yeah.
Yeah. So go back to the other one. Say A had 4 there.
Uh-huh.
Would it do the same thing as A and B in this?
It would be. That's why when you form more synapses, it makes you more effective at communicating with the postsynaptic cell, right? Because it doesn't matter what this is attached to ultimately, right?
Okay, cool.
That makes sense? So yes, so this reinforces the idea of synaptic plasticity. If you really want the postsynaptic cell to be responsive, you make more synapses to the postsynaptic cell, right? And then if you want it, we're going to get it started fast, you're going to get it started faster now. That makes sense? Okay, so this is called spatial summation. This is how you get this postsynaptic cell to integrate all of the inputs and come up with an output. Okay, now let's briefly, before we get into the specific neurotransmitters, just talk theoretically about, okay, I want to just theoretically say, look at this particular synapse and say, okay, I've got a problem with this synapse. Either I am releasing too much of a neurotransmitter or I'm releasing too little of a neurotransmitter, okay? What, with our current level of knowledge, can we do in a situation like that? What can we do if you're releasing too much? What can we do if you're releasing too little? Well, ideally, what we would be able to do is to say, hey, you are not making enough of it, make more. You are making too little of it, make less. We have almost zero capacity to do that, FYI. Okay, so depressed or anxious might be low levels of serotonin, make more serotonin. Can't really make you make more serotonin. We can tell you all of the things that might increase your serotonin. Exercise more, meditate, eat some good food, have a little more sex, and then hope for the best, right? And sometimes it works, but sometimes it doesn't, right? So we have almost zero capacity to really tightly manipulate the level of neurotransmitters that you're making or not making. Schizophrenia, too much dopamine in portions of the cerebral cortex. How do we get you to make less? So we can't really do that at this level. Hopefully at some point we will be able to. So that one's not so possible yet. But what we often do instead is interfere with what's happening in the synapse after it's already occurred, okay? Because a lot of biochemical research and protein research has gone into knowing the shapes of these, and these, okay? So what we quite often do is we interfere with the removal of the neurotransmitter after it's already in the synapse, right? Because we know a whole bunch of reuptake mechanisms. We know some breakdown mechanisms, right? That's primarily what we do. So let's say you go in and Utah, the last time I checked, and I think it's still true, it was true all but one of the last 10 years that Utah had the highest per capita antidepressant and anti-anxiety use in the country. Let's say you got depression or anxiety. Those are quite often, but not always, related to low levels of serotonin, okay? And you want to mimic the impact of higher levels of serotonin so that you get release from depression and anxiety. One of the major treatments that they use, and the first tier of treatment that they use, is a selective serotonin reuptake inhibitor. And what it does is, if serotonin is your mood regulating chemical, which allows you to basically handle what life throws at you without completely having a meltdown or going into a cave. So what it will do is, for every time serotonin is released, instead of immediately reuptaking it, it lets it sit around longer. And that really mimics releasing more serotonin, right? Because the postsynaptic cell doesn't know whether you released it once and let it stay or release it frequently. Does that make sense? Okay. Now, interestingly, for those of us, and I know there are a bunch of us here in this room who are crazy like me and have tried just every damn crazy medication to see if you can control your crazy. Do they ever test your serotonin level before they give you an SSRI? Why not?
It's in that little spot.
Yeah, it doesn't go into the bloodstream, really, right? It just goes in here and it's immediately removed. So what are they doing? They're just like experimenting with you, right? Just like take this and see if you feel better and, you know, please drive through. But you can be involved in the experimentation process, of course, right? So if you take any neuroactive chemical and you really want to see it, whether it works, I have a spreadsheet. When I took it, how I slept, how much sex I had, whether I was pissed off at my kid, all that for like a month before I go back in, you know, and then I'm like, I don't like this one, or I do like this one, or we need to half the dose or up the dose or whatever, because I'm like, you know, apparently I know more than you do. At least about my brain I do, right? SSRIs are really a common way. There's the other, we'll talk about this one too. The other next tier, they don't usually use it before they use SSRIs. They can use an SDRI and an SNRI. That's Wellbutrin. Wellbutrin is bupropion. It's a selective dopamine reuptake inhibitor and selective norepinephrine reuptake inhibitor. It allows those two to stay in the synapse a little longer. And so those are really common. MDMA, which we will talk about, is ecstasy. Ecstasy does two things related to this story. It's both a reuptake inhibitor for serotonin, and it also causes a massive dump of serotonin. So a massive dump and inhibits reuptake simultaneously. It was also just approved by the FDA for a third level trial for PTSD. And it's been really super duper duper effective for PTSD. I'll teach you guys about MDMA later. I'm not talking about party MDMA. I'm talking about like medical grade MDMA. Okay, so we'll talk about these as we go through. So the other thing that you can do, and they do, is interfere with binding to the receptor. So you can interfere with this. You can also interfere if there's an enzyme. You can block the enzyme that breaks it down, and we'll talk about those. But you could also interfere with binding to the receptor. So there's two different classes of drugs that do this. There are agonist drugs and antagonist drugs. An antagonist drug sits in the receptor and prevents the postsynaptic cell from having the response. They call those blockers. Yeah? And the agonist drug sits in the receptor and causes a similar response on the postsynaptic cell. That's like a mimic. Okay? They're usually not exact, but they can get you some kind of similar response to the postsynaptic cell. That makes sense? Okay, so let's talk about a couple of agonist and antagonist situations. So with an agonist drug, produce a response that is similar to the normal activation of the receptor. Part of the reason that people like nicotine is because the initial feeling of nicotine is because of its agonist effect. Nicotine is an acetylcholine agonist in the central nervous system, and an acetylcholine in the CNS causes an increase in short-term mental acuity and short-term memory formation. So can you see why it might be rather pleasant in the initial stages? Any idea? Yeah. Unfortunately, when you do that over a long period of time, what's going to happen to your receptors? You're going to downregulate your receptors. And then will you get the response? No, you're not going to get the pleasant response that started the whole thing. You will have, however, have tapped into the habit formation pathway. Go ahead.
So what does the medical industry do to address that with SSRIs, for example? I mean, wouldn't you down-regulate taking whatever it is?
Well, what you try to do is to always, first of all, you need to go to a well-educated medical professional, and you always take the lowest dosage you can possibly take, because the downregulation occurs with large quantities. If you can keep the amount in the synapse to relatively close to normal quantities, then you have less downregulation, right?
But do
so,
you always have downregulation?
Yes, you always do. That's one of the many, many reasons why when people are trying to get off of SSRIs, you can't go cold turkey off of SSRIs, because you need to have time for your upregulation to occur. So you said always. The answer when there's an always question is probably not, but almost every situation that I've ever heard of, you will have downregulation in those. So you always take the lowest dosage that could possibly work for you. Okay, so nicotine is an acetylcholine agonist. Okay, antagonist drugs or chemicals you're going to run into when you are, for instance, dealing with like having too much of something, or antagonist drugs or chemicals are often used as blockers. So here's an example. It's not neurotransmitter, it's endocrine related. If you have a dude who is deciding he's going to pump up by taking testosterone, one of the things that happens with excess testosterone is that it gets converted into estrogen. So if he wants bigger pecs but not bigger boobs, he should probably take a what antagonist? Estrogen antagonist. They call them estrogen blockers. The same thing goes with people who are transitioning, right? People who are transgender and they are transitioning from female to male, right? Usually even before they start taking testosterone, they take estrogen blockers, estrogen antagonists. That makes sense? To block the response. Because you can't really make your body stop making it initially, right? You just got to block the response of it, okay? But the other instances of antagonist drugs or chemicals, this one's kind of interesting. So caffeine. Let me teach you how caffeine works on your brain. It does stuff to your peripheral nervous system too, but let's concentrate on the brain stuff. Caffeine is an antagonist to an inhibitory neurotransmitter. So what do you get? You get excitation. You get CNS excitation by blocking the response to an inhibitory neurotransmitter in your brain. Does that make sense? That's how that works. It does other stuff here. The peripheral nervous system also increases your heart rate, makes you pee, and do all kinds of stuff. But the CNS activity, the reason it wakes you up is because it blocks the effect of an inhibitory neurotransmitter.
What is it?
The inhibitory neurotransmitter is hold on, I forgot which one it is. It is adenosine. Okay, so, all right. So the other times you're going to run into, antagonist drugs or chemicals are usually with poisons. And they weren't like, they didn't evolve to poison you, but they're a defense mechanism in an organism to make sure that if something eats it, it's not going to eat it twice. And if it lives through it, it's going to tell all its little buddies or die in front of its buddies so that you're not supposed to eat that brightly colored whatever, right? And quite often, a good way to kill people is to block a neurotransmitter from causing its effect, either on cardiac muscle or skeletal muscle or between neurons, right? So antagonist drugs or chemicals are very often poisons. One of them is tetrodotoxin from the kilo pufferfish. Look at the kilo pufferfish. He will kill you dead. So this is a pufferfish. And it has a little gland that's full of a toxin called tetrodotoxin. And what tetrodotoxin does is it's an antagonist to a neurotransmitter and allows you to have complete respiratory failure while you're completely awake and aware that it's going on. It blocks the response of your neurotransmitters on your skeletal muscle, including your diaphragm. And so you have conscious respiratory failure. And of course, because people are really, really bored, what do we do with this one? We eat it, yeah, and we charge like $500 a plate for it. I've never had it because it's too expensive, but I'm totally gonna. So, huh?
Which part is the fish?
All of these little slices all the way out here. Super duper, like delicate. It's supposed to be, has anybody tried it? It's supposed to be delicious. In addition, when you eat it, you get a little tingle that means you almost died, but you totally didn't. And you were not bored at that particular moment, were you? No, you weren't. So it has to be really carefully prepared. You can't even get it in this state. You can get it in California, and of course you can get it in Japan. If the chef preparing it does not prepare it properly, you will die from eating it. And it's outrageously expensive because of the training needed to prepare it. And I'm totally going to try it one day. We went to Japan, and I was like trying to get my husband to buy it, and he's like, you can pay for crap like that with your own money. I'm like, damn it, I don't want to pay for it with my own money. What? Next time I go to Japan. I was going to like say, why one of you guys make it for me, but I forget that I'm like, I've got your grades and you might kill me. Okay, so that's puffer fish. Have you guys heard of red tide? Anybody heard of red tide? This is a protozoan that under certain environmental conditions gets big algal bloom. It's not algae. It's a little protozoan. Big blooms in the ocean, and when that occurs, it's basically a neurotoxin, and it gets into all of the sea life, and you can't eat a damn thing out of there, and you're not supposed to swim because if you accidentally took any via your mucous membranes, you could die because of that. So that's red tide. It's also a neurotoxin. And then there's a couple others. Black mambas, they'll kill you. Did you know that? Yeah, they will. That's because their poison, their venom, is a neurotoxin that's an antagonist. These two won't usually kill you because they stay local. So honeybee venom is an antagonist, but it doesn't usually go systemic. So it's usually just going to cause problems at that location. Same thing with sea anemone. Okay, so now let's start getting into neurotransmitters. You ready? Okay, so neurotransmitters, we are not going to learn all of them. So how did I pick which ones I'm going to teach you? Well, considering you guys are going into stuff related to human health, I want to teach you anything that you need to understand the rest of the semester, and then anything I think you've either popped in your head hole or you're going to treat somebody who's popped in their head hole, right? Or stuck in a vein. I don't know what you're going to do with it. But the ones that you are going to run into, either with your own medical care or the medical care of other people, are the ones that I'm going to try to teach you. There are at least 60 neurotransmitters, and we haven't even discovered them all. And neurotransmitters can vaguely be categorized as CNS neurotransmitters or PNS neurotransmitters. Which one do you think we know far more about? PNS neurotransmitters. You can do experimentation with PNS neurotransmitters. I can, for instance, zap myself and make myself release acetylcholine without causing any brain problems, right? You can also use model organisms to figure out that kind of stuff. But which ones are more interesting? The CNS ones. But we don't have a good model organism for human higher functions. Like, how does a rat manifest anxiety? I don't know, it pulls its whiskers out, but can it really tell you how it feels about its mother? No, it cannot, right? So the CNS ones are super duper interesting, but a lot of times the information we have about CNS neurotransmitters in humans is somewhat anecdotal. So how do you figure out why somebody has schizophrenia? Well, you don't give them schizophrenia and then figure out what the hell happened. You have someone with schizophrenia, you do fMRIs, you do all kinds of testing to figure out if you can tell what's different between them and someone else who doesn't have schizophrenia. So there's a lot of holes in our knowledge, but there's some that have gotten filled in. So I'm going to teach you about the neurotransmitters that I understand well enough to talk about. In addition, because you guys are curious little souls, you're going to have a whole bunch of questions about drugs either did, wanted to do, or want to judge somebody else because they did. And they're all down here at the bottom. Not all of them. This is a very small proportion of the questions that I get all the time. So you can, I want to know how cocaine works, because that one friend that I had that tried cocaine that one time, then you want to know which neurotransmitter it works with. Alcohol, that kind of stuff. Now, this is only a small portion. I have like an entire spreadsheet that's like this big of all of the drug actions and brain actions of every kind of drug that I could ever find, including the ones that are illicit, illegal, and the ones that aren't even considered drugs but have neuroactive effects on the brain, like sugar, like omega-3 fatty acids, like all of that kind of stuff, because I collect the stuff. That I'm like a facts curator, that I just like make spreadsheets on my spare time. And that's why I have 3 friends, all of them. And they're just like, we're going to just hang out with you for fun. Okay, so we are going to get started on our neurotransmitters. So you guys tell me before we go any further, in order for something to be considered a neurotransmitter, it has to meet 2 criteria. It has to be released from a neuron close to a receptor. It doesn't have to be a neuron, but it has to be a receptor. It could be a muscle, or right? Yeah. Okay.
Because
so
it doesn't go through the blood.
It does not go through the bloodstream.
But it's because it has to be close to it because there is...
Right, because there's no other way for it to travel. Yeah, got it? Okay, so they can be CNS or PNS, yeah? And some of them are both, right? For instance, you use acetylcholine in the brain, but you also use it with skeletal muscle, okay? So when possible, I will tell you the CNS function and the PNS function if I know them. And usually their mechanism is to open an ion channel either directly with an ionotropic receptor or indirectly. I don't care if it's indirectly, it just means it's going to be a little slower, right? So usually they will either depolarize or hyperpolarize the postsynaptic cell. 60 of them that have been identified, we're finding more all the time. They just figured out about 15 years ago that nitric oxide, nitrous is the giggle gas, nitric is another usually gaseous thing, and they figured out that it was really important because it's actually involved in erections. And no matter how much money is required, they are going to study erections, right? It's like all of the money that you have to study erections. So yes, a lot of the drugs that they use as erectile dysfunction drugs use that neurotransmitter. Also, ATP is a neurotransmitter. Carbon monoxide, the hell? That'll kill you if you breathe it in. It's actually released as a neurotransmitter. So I'm not teaching you 60. I'm going to teach you the ones that I think you're going to run into. And then you're going to realize that you do not have a complete understanding of all of them. Okay, so what is a neurotransmitter usually like? They're pretty small, meaning we're not talking about enormous proteins. Usually they're about the size of an amino acid, okay? Sometimes a few amino acids stuck together. The biggest neurotransmitters are going to be the slowest to diffuse across the synapse, right? Okay. The biggest one that I know of that is, I won't even teach you its function, is 40 amino acids big. And that's not really even a huge protein, right? But most of them are relatively smaller than that. So since most of them are small, they're relatively fast acting, and they quite often there are more than one receptor for a neurotransmitter, meaning that it's fine if you want to say that acetylcholine does this, but it might do something else in a different tissue because it might have a different receptor. Because really the cell's response is more about the receptor than it is about the neurotransmitter, right? Isn't it? Okay. So what we are going to do is we're going to start categorizing them, and we have to categorize them first based on their chemistry. And this isn't like hard chemistry. Don't worry about it. It's just that the reason you will have to understand a little bit about their chemistry is if 2 things are very, very chemically similar, like these guys right here, they sometimes use the same reuptake mechanism or the same breakdown mechanism, and sometimes you can use drugs to treat them all at the same time if they're really similar to one another. So that's why it's worth it for you to figure out like how they're categorized, okay? So here's what we're gonna do. We are going to organize these neurotransmitters. We're gonna say, okay, they're all neurotransmitters, and we're gonna categorize them. The first category are the choline derivatives. And they are derived from a water-soluble B vitamin called choline. That's what you use to make them. And really, the only one that you're going to need to learn from here is acetylcholine. There are others, but the acetylcholine is going to be the biggest one. Okay, so I'll show you what acetylcholine looks like, even though you don't care. And no, you never have to draw this chemistry for me. Okay, but can you see that it's not enormous? Right? These are carbons. If you haven't had organic chemistry, everywhere you've got a point as a carbon. So it's not outrageously big, okay? So acetylcholine is the one that we know the most about because you use acetylcholine between every single neuron that communicates with a skeletal muscle cell. That means that you get a lot of data, right? Every single skeletal muscle cell is going to use acetylcholine as its neurotransmitter. So you can mess with it in humans without killing them. You can do it in any mammal. I know it's in all birds, used in all birds, and I think it's in all reptiles and amphibians. So it's like really broadly used. So it was the first one we learned about. It's the best understood one that we have. It is usually excitatory, and I'm not saying that because I don't know, but in most situations it binds to like a sodium ion channel and causes depolarization. And its CNS function, we already talked about a little bit. Its CNS function is you use it for short-term memory formation in some of the deeper portions of your brain prior to long-term memory formation. And one of the reasons we know about that is because what happens in Alzheimer's, which is right down here, we'll talk about it right now though, Alzheimer's disease represents a 90% loss of acetylcholine in certain short-term memory formation portions of your brain. Here's what we don't know yet though. We don't know whether the glial cells stopped supporting the neurons, and that's why it went bad. We don't know whether you stopped making acetylcholine and therefore lost the receptors. We don't know whether you lost the receptors first and then stopped making acetylcholine. So these are areas of research that are going on with CNS functions and acetylcholine. Good? Okay. Now the PNS functions of acetylcholine is every single skeletal muscle is going to use acetylcholine as its neurotransmitter. So the entire somatic nervous system, does that sound familiar, somatic nervous system? The entire somatic nervous system uses acetylcholines, and about half of the autonomic nervous system uses acetylcholine. That make sense? Okay. So let's look at acetylcholine synapse real quick. Oh, not that one. Okay, so there is a short way of saying this. This is all releasing, responding to, using, reuptaking, all of that. The whole thing is acetylcholine. So the short way to say that is just to call the whole damn thing cholinergic. That's a quick way of saying everything related to acetylcholine. So this whole synapse is cholinergic. The receptors are cholinergic. The reuptake mechanism is cholinergic. That makes sense? Okay, so you call it cholinergic. Okay, so everything would be considered cholinergic there. Now, as far as receptors for acetylcholine, the receptors would also be cholinergic. And there's two types based on how we found them. There is nicotinic, cholinergic, and muscarinic. Any idea how we found those? Yeah, gave somebody labeled nicotine, watched where it went. Those are nicotinic cholinergic receptors. Yeah. And then muscarinic, this is a mushroom poison that kept killing things. And we wanted to know why it was killing things. So they labeled it and let it kill things and see where it went. Mushroom poison, muscarinic. Okay, so we'll run into nicotinic and muscarinic on like skeletal muscle and cardiac muscle as we go through the semester. Good? Okay, so nicotinic and muscarinic. Okay, so just a little bit before we move on. As we mentioned, nicotine is an acetylcholine agonist in the CNS. Yeah? And it's also lipid soluble, so when you take it in, you get that really quick response, right? Short-term mental acuity and all that kind of thing. Have you guys ever heard of curare? Curare is a poison. Oh, you have? Good. Somebody read mystery novels as a kid besides me. Oh, it was Batman. Good. Whatever. Mine was in Agatha Christie novels when I was a kid. So curare is a poison. It is South Central America. I'm terrible at geography, so I'm always lying when I say anything geographic. I'm just like trying to get on the right continent, right? Down there, okay, pretty sure it's like on the southern continent, South American continent. And it is derived from a plant and has been used by indigenous people for ages and ages. And what it does is it will paralyze and block your acetylcholine receptors on your skeletal muscle. So let's say you want to eat something that's in a tree and you don't want to climb the tree. You put it on a blow dart on an arrow, and then you shoot the something, And then eventually the diaphragm will paralyze and the muscles will paralyze and they just fall out of the tree and then you can eat it. Or if you want to take down some large game animal but you don't want to chase them, you just like shoot it at them and then just wait for them to fall over. Okay. Andre brought up a good point though last time. He's like, well, why the hell would you like eat something that you just poisoned? Has a really, really short half-life. So it degrades really quickly. I don't know how quickly, right? Apparently they know. So that's curare. And of course, since we've been using it to kill animals, of course we use it to kill people because people don't like people very much. Have you ever noticed? Not so much. Okay. So that is acetylcholine. We will run into acetylcholine a whole bunch this semester. Okay, and then now the next category, I think are maybe the most interesting. These are called the biogenic amines. So let's just write down the name and then I'll explain what I mean by biogenic amines. Okay. Take an amino acid. Okay. You guys remember amino acids? You don't, do you? Okay. I don't care whether you remember what they all look like, but do you remember that an amino acid generally has this pattern of C double bond OH and then a nitrogen containing group on one side, right? And then this is the R group that changes however the hell it wants, okay? If I took one of these 20 amino acids and changed it just a teeny bit so that you couldn't use it as part of a protein anymore, but it was still pretty much the size of an amino acid, then you've made it into a biogenic amine. So let me show you. And these are the ones you're about to learn anyway. And no, you don't have to memorize the structure. Here's tyrosine, which is an amino acid. So is this one, though. Take tyrosine, look at it. Change it just a teeny, teeny bit right there. It becomes something else. Change it just a teeny, teeny bit more right there. Yeah? Then it becomes dopamine. Yeah? Change dopamine just a teeny, teeny bit more. Where is the dopamine? And it becomes, where did I do it? Here. Put a hydroxyl group on it. It becomes norepinephrine. And then change it just a little bit more and put a methyl group over here. And then it becomes epinephrine. So why the hell do you care? These three are so similar that you will see them using the same reuptake mechanisms. They can be medicated with the same classes of drugs. That makes sense? Sometimes the same breakdown mechanisms. So it's important for you to know that those are really, really similar to one another, okay? So the first set of the biogenic amines that are about the same size but a little bit different than a straight amino acid are going to be these guys, which are called the catecholamines. And those are the three that you just saw, which is dopamine, and then norepinephrine and epinephrine I always put together because they almost always interact with the same receptors and the same reuptake mechanism. So you can kind of lump them. But I'll tell you about them in just a second. Okay, so dopamine. Dopamine is probably your favorite neurotransmitter. You just didn't know you had a favorite. But dopamine is like the source of all things pleasurable that you want to repeat. Here's what dopamine does for you. Dopamine is reward, reinforcement, motivation, perseveration. When you just did something and it was awesome and you think, I can't wait until I can do that again, that's dopamine, right? So evolutionarily speaking, you can see that it's probably really pleasant if you want to reward yourself for something, right? So evolutionarily speaking, you can imagine that you would release this when you ate something that was really nutritious, like sugary or fatty, densely nutritious. When you have sex, you release dopamine. Some people must release a whole hell of a lot of it when they exercise, but I don't think mine works that way. Mine releases whatever when the hell is this going to be over neurotransmitter part. So reward, motivation. So it is motivation, reinforcement, and reward. So food and sex, but then wire in drugs in there. If you want a drug to be really pleasant, dopamine is going to make that drug really pleasant. It will also make it very, very likely to reinforce the behavior. Can you see that? That the drugs of habit formation and addiction, almost all of them tap in either directly or indirectly to the dopamine pathway. So let's brainstorm for a second. Drugs that are truly addictive. Not just yes, all of the opioids. The opioids, what? Alcohol taps in a little bit, but it seems to actually be really impacted by your genetics, how much dopamine it actually releases. What other ones? Meth, cocaine, yes? Almost any amphetamine. Now, the problem is that sometimes you're using them for another reason, right? But then you tap into the dopamine pathway. So you have an injury, and then you go in and you're prescribed opioids, yes? Because opioids are massive pain relievers. They're the most effective pain relievers that we've found, especially for acute situations. But then you take them, and then what happens is you tap into the dopamine pathway. And then here's the problem with tapping into the dopamine pathway, whether you're taking an illegal drug or a legal drug, whether you're taking it for illicit purposes or for medication purposes, is that once you start taking it, what's going to happen to your receptors? You're going to downregulate your receptors, yes? And then what is it going to take in order for you to have the same level of reward? More, yes. More dopamine release, right? Yeah, it's a totally vicious cycle. And other countries don't really use opioids in the way that the United States does. So we're probably going to have some conversations during this class about like what you think about the future of pain management and what things we might want to consider, because you guys will be voting, right? When it comes next time they put marijuana on the ballot and you think about marijuana versus opioids, then you might want to recategorize those treatments for chronic pain. Go ahead. I'll teach you how marijuana works too. Go ahead. Yeah, we'll talk about it. It's a really interesting one. It works totally different than any of the other ones, and it doesn't tap into the dopamine pathway at all. It doesn't. I'm not saying it doesn't. People do not form habits with using marijuana, but it does not tap into the dopamine pathway. It has no dopamine response at all. Go ahead. Oh, yeah. There's like behaviors that you can do that will tap into the dopamine pathway too. Gambling, social media, sex, anything that really taps into it, and then you start reinforcing it, and then you take it away, then you like get a dopamine crash called anhedonia. So let's talk just briefly about dopamine and addiction and tell you some of the research that's going on that is not confirmed yet, but some interesting questions that you can go out and read some about. When you are reading about addiction, please go to .edu sites. Do not go to David Avocado's Wolf site because that dude's an idiot. And don't go to your grandma's site because she's really, really nice, but she didn't know about this. .edu, .gov, you can go to those sites. So one of the things that's being studied right now is why the hell are some people so predisposed to addiction, right, regardless of the kind of addiction compared to others. Some of you guys may be from addictive families. I am from an addictive family. Both of my parents are dead because of addictive behaviors. My older brother is a recovering addict. I am literally the only adult who does not have addictive behaviors. Although I probably do. It's like my spreadsheets or like my obsessive whatevers that I do. I'm just not shooting anything up. So what is that about? One of the things that they are studying, which will be really interesting to find out, is they think that it might have something to do with your inherent level of dopamine release with regular behaviors in life. So let's say I'm cuddling with my kid and I am watching The Office, right? And I'm having a milkshake. And if my level of dopamine is like pretty high, then when I decide that maybe I'll do meth, is it going to be like as dramatic a difference between my relatively high level of dopamine inherently and the level that I get from doing meth? Whereas with someone who has relatively low levels of dopamine inherently, when you actually tap into the dopamine pathway with some kind of habit formation thing, the response they think is really major. And then the crash afterward is really big, right? So then you get the downregulation of the behavior, and then you get the perseveration and the seeking out. So what they're hoping is if they can actually confirm this, then you could actually like go in and get tested when you're an adolescent and you're seeking behaviors, right? When you're in your experimental phase. You're going to go in and take a test to see if you should try crack. You should probably not try crack. But I mean, it's just a good general policy. But some of us should really not try crack, right? Just really not. That makes sense? So they think it might have something to do with your inherent level of dopamine in this particular part of your brain called the nucleus accumbens. And your nucleus accumbens is really, really responsive to this dopamine reward system. So it's kind of interesting. And you will see the addiction behavior manifests in multiple ways. Like if a person is predisposed to addiction, it doesn't have to be a chemical addiction. It can be a behavior addiction. It can be working out. It can be sex. It can be gambling. It can be almost anything, right? Go ahead.
So we've talked about synthetically inducing a downregulation. Is there a way we can do an upregulation on things?
Not yet.
Not yet.
Not with CNS stuff. We still have so little that we know about the like inherent levels, and then you'd have to have a model organism to experiment on. You know what I mean?
Yeah.
Like even if I thought it was gonna like, okay, I'm gonna give you this medication, and it could either make your IQ point go up 20 points, and you're gonna lose 30 pounds and look 25 years younger, or you're gonna crap yourself and you're gonna have an IQ of 10 for the rest of your life. Any volunteers? No, that's what I thought. So the problem is we don't have a good model organism for higher level human functions. Go ahead.
With dopamine responses like addiction, can you get a crash? Like not just go back to your normal levels, but like dip below your regular.
You will after an addictive cycle, you will dip below your normal levels, and that's called anhedonia. When you like no longer take any pleasure in normal behaviors, and a lot of times it's temporary. Anhedonia. Hedonism, but the opposite of it. And
Oh, I see. It's like withdrawal.
not Annie. Annie doesn't have anything to do with it. Yeah. The withdrawal is bigger than that. That's just like the mental effect of the withdrawal. The withdrawal has all kinds of physical effects as well. Okay, there is another thing that dopamine does in your brain that is unrelated to this habit formation, reward, and reinforcement. So I want you to separate it because these two don't have anything to do with each other. There's a different portion of your brain called the substantia nigra. Deep in your brain, it's one of those cerebral nuclei that's way like in there far. And the substantia nigra controls the pathway for voluntary motor activity. And when someone has Parkinson's disease, they start losing dopamine in that area of their brain, and they lose control of their voluntary motor activity. But it's not related to the other dopamine, right? It's a totally different portion of the brain that uses the same neurotransmitter. That makes sense? So I'm not saying that people with Parkinson's are junkies, or junkies are going to get Parkinson's, right? Right, not necessarily related at all. Two different places in the brain. Good? Okay. So, schizophrenia, last one. Schizophrenia, really, really difficult to treat. But one of the things that we do know about schizophrenia is that it is releasing entirely too much dopamine in specific portions of the cerebral cortex. It's really, really difficult to treat properly. Does it make sense to you that it's harder to treat a disease that causes a hyper secretion than it is to treat one that has too little? So what the hell do you do? You might be able to use an antagonist, but it has to be an antagonist that's just specific to there because of, right? Because if you use it elsewhere, you get anhedonia or muscle tremors, right? So you have to know an awful lot about the receptors that you're dealing with before you can actually decide to use that. So lots of theoretical things, but there's an awful lot of research that needs to go in. Go ahead.
Drug use that affects the dopamine pathway would likely act as a catalyst for their schizophrenia.
Yes.
Because you'd have exacerbated downregulation.
And as a matter of fact, that is true, although we do not know the specific mechanism yet. Sometimes drugs of abuse will trigger schizophrenia when you had the predisposition to schizophrenia. Schizophrenia has a really strong genetic component, right? But it also seems to have quite often environmental triggers. And so if you really wanted to turn your schizophrenia on, you could use some drugs of addiction. You can also use the serotonin drugs, the hallucinogenic drugs, to seem to be able to do it too, even though they're not directly related to dopamine. Okay, now let's talk about these guys. It's not dopamine, it's dopamine. By the way, dopamine, last thing, dopamine receptors, the receptors are just called dopaminergic. And there's D1 and D2. And I think, is it D2? I think it's the D2 receptors that are ones really, really implicated in addiction. Some of the drugs that they use to get people off of addictive drugs will block those receptors. So there's some drugs that we'll talk about down at the bottom too, at the page. let me show you. See if I wrote that down. Somebody asked me about this last time and I had to find it. So the opioids don't directly work on the dopamine pathway, but they tap into it. And the dopamine drugs, D1 and D2, if I remember correctly, it's D2 that is really, really involved in reward and reinforcement. And if you can block that response with like some of the drugs that they use to get people off of heroin, then you can block the addiction response during the moment, or you can make it really unpleasant for them, like the opposite of hedonia. You can give it like really unpleasant. Okay, so we'll talk about that too when we get back to the opioids. All right, so let us do norepinephrine and epinephrine. Okay, so norepinephrine and epinephrine kind of go together because they're super duper closely related to one another. But this one is usually CNS. This one can be CNS or PNS. Okay, and there's overlap between both of them, but mostly they interact with the same receptors, so you can really lump them together. Okay, so let's talk about those. Let me remind you what they look like. Look how close they are to one another. The difference between them is simply a methyl group sitting off right there. So if the binding site was over here, then they would interact with pretty much the same receptors. Yeah? Okay, so their function, going back to our little Venn diagram of happiness right here, their function, norepinephrine and epinephrine, is basically they are your up neurotransmitters in the brain. Alertness, concentration, and energy in the central nervous system. So if you have all three of these and you get into the sweet spot of the Venn diagram right there, everything is firing high levels and you're super duper happy and able to handle anything. Has anybody ever been there? Like maybe 10 minutes, my entire life right there. Right? It was great. It's good 10 minutes. Loved it so much. So norepinephrine and epinephrine. Now, alertness, concentration, and energy. And in the peripheral nervous system, these are the ones that are involved in sympathetic nervous system response, the fight or flight response. Okay, that's a PNS response though. So I'll tell you a little bit more about those in just a second. Any cell that has receptors for and responds to norepinephrine and epinephrine, these are all catecholamines, by the way. These are going to be called adrenergic, not norepinephrine or epinephrinergic, because when we found epinephrine first, we found it in the bloodstream because it's also released as a hormone from what gland? Adrenal gland. Then they tracked it back to the adrenal gland and they named everything related to it adrenergic, and then they just never changed the names once they figured out that they came from also neurons. Okay, adrenergic. And then the adrenergic ones are just alpha and beta receptors. That's it. We'll find those beta receptors later. Okay, so catecholamine removal. Okay, so basically, let's go back to this. These are all catecholamines, these three. So let me imagine right here that this is a catecholamine receptor, right, for dopamine, norepinephrine, or epinephrine. They usually use the same removal mechanism, so you can treat them together a lot of times. So this is a reuptake mechanism, and this is a breakdown mechanism. So let me introduce you to those. So catecholamine removal, there is a reuptake mechanism at some synapses, and at the other synapses, there is a breakdown enzyme called monoamine oxidase. Okay, which means that we could treat dopamine, norepinephrine, and epinephrine all at the same time, quite often. And you do, and you may have, because Wellbutrin, or bupropion, is another way of treating depression. If the SSRIs didn't work for you, you could use Wellbutrin. SSRIs will work on it this way. Where's my Venn diagram? Okay, an SSRI would work on it this way. So you would try to regulate your mood. If that failed, you could use an SNRI and an SDRI, which is Wellbutrin or bupropion. And what those do is allow those two to stay in the synapse a little longer with one medication, because the chemicals are so similar to each other. So you can use one medication for them. Now, it feels different to take these two drugs. I've taken both of them. The SSRIs, the symptoms associated with it is kind of a flat affect sometimes, right? Like you don't really get anxious about anything or particularly depressed about anything, but you also don't get anything about anything, quite often, right? A lot of times you can gain 30 or 40 pounds, completely tanks your sex drive, okay? And sometimes sleep disturbances. But, you know, if you were almost suicidal before you took it, that might very well be a good bargain. There are a whole bunch of SSRIs to choose from. We will talk about them next time, but here are the SSRIs that are available right now. But Wellbutrin or bupropion works differently. It can still make you happier and pull you out of depression, but it will feel different, won't it? Won't it feel slightly happier and more rewarding and slightly upper? That's a word. Just made it up. Okay, so it's going to feel different. You don't gain 30 pounds. It doesn't trash your sex drive. But how well do you think it would work for anxiety?
Not good.
No, it is not a good anxiety medication at all. Like if you were anxious, you're going to get extra super anxious. If you had panic attacks, that is not the medication for you. This is one of the reasons why you need a little background in order to figure out how to tell your doctor what would potentially help you. So this is how Wellbutrin works. It works on those two by using one medication. So Wellbutrin is norepinephrine and dopamine reuptake inhibitor used as an antidepressant. It's not indicated for anti-anxiety though. And the last one is one that's hardly ever used even though it's pretty damn effective. And this is monoamine oxidase inhibitors. So it's going to do the same thing. It's going to work at different synapses. So this time I want you to imagine that the synapse is going to use a breakdown enzyme called monoamine oxidase, yeah? What I'm going to do is inhibit monoamine oxidase so that it can stay in the synapse longer. And you're still going to get that up, right, and happy response, okay? So why don't they use that? Those two are called Nardil and Marplan. Does anybody work in psych? You do?
I did a month ago.
Oh, you did a month ago? I'm always curious about these because they hardly ever use them anymore. Huh?
I worked in psych for 4 years.
Oh, you did. So Nardil and Marplan, they're actually pretty good from what I've read, and maybe he's got some help with this. Really good for treatment-resistant depressions, but they interact with damn near everything known to man, so you can almost only use them in an inpatient situation. Because, like, you can't have cheese with them. I'll be depressed just because I can't have cheese, man. You can't have wine with them. Like, that's, like, literally half of the things I look forward to in life, wine and cheese. There's a whole bunch of over-the-counter medications that you can't take with them. There's a bunch of supplements that you can't take with them. There are a bunch of things that you can't eat with them. So basically, you would have to have somebody who was really, really tightly controlled about their behavior. So they hardly ever use them anymore, even though they're effective. Did you ever have anybody that you knew was on either of those? Yeah, I don't know how often they're used. I know that they are still in the suite of medications that you could choose, but as soon as somebody is released, they're not going to be able to be on them any longer, because the responses when you interact with other things is pretty violent. Also kinds of nervous system disturbances and complete like throwing up and all that kind of stuff. So that's Nardil and Marplan. I want to know if anybody knows the situations in which they're still used. Okay, and then, Okay, good. Okay, so the other one with our last 10 minutes is talking about serotonin. So these are all catecholamines, and they go under biogenic amines. Serotonin goes by itself. And I'll tell. Oh, right. And these are derived from tryptophan.
Wait, so serotonin is a biogenic amine?
It's a biogenic amine, but not a catecholamine.
But
Okay.
those three are.
Those three are catecholamines. 1, 2, 3. Okay. And then serotonin is a biogenic amine, but not a catecholamine. Catecholamines are all derived from tryptophan. Let me show you the catecholamines again so you can figure out why the hell they go together. And by the way, this gets easier. So here are all of the catecholamines. They're all together because they all have the same mechanism, or basically the same precursor. And serotonin is derived from tryptophan. It's basically 5-hydroxytryptophan. Okay.
So both catecholamine and serotonin comes from tryptophan.
Oh, did I say tryptophan? I didn't mean tryptophan.
It's tyrosine.
Tyrosine. Thanks. I was testing you. You did great. Okay. That was derived from tyrosine. Serotonin is derived from tryptophan. It's just 5-hydroxytryptophan, 5-HT. You can buy that stuff over the counter. Whether it works or not, I don't know. Probably shouldn't, but might make you feel better. Okay, so this one is off by itself, and sometimes it's called indolamine. It is quite often called 5-HT. So what are some functions? Just listen to all the damn functions of serotonin. Okay, what does serotonin do in your CNS? It's important in the entire brain, especially in the brainstem, meaning the brainstem is really going to do processing of input and output of sensory and motor, so keep that in mind. It's usually inhibitory in the brain. It regulates sleep, emotion, sensory function, motor function, hunger, mood, sex drive, anxiety, obsession, compulsion, and memory. Just a few things. Okay, so when you mess with serotonin, you better be selective. Yes? You want a selective serotonin reuptake inhibitor. You do not want one that impacts your memory. Yeah? You do not want one that impacts your motor function. You do not want one that impacts your sex drive if you can avoid it, right? But how selective are they? Well, they have gotten more selective as time goes on. So as time goes on, some of these newer ones, you can see the first one was Prozac. Zoloft was not the last one created. It's right back there, right? So number 12 is Paxil. Lexapro is also number 12. So these are some of the most, the newest ones. So these are used for treating depression and anxiety. And typically speaking, now this is an overgeneralization, the older they are, the less selective they are and the more symptoms, side effects you'll get with them. Also, the more robust they are, the more people that they will work in, okay? So, but you might very well lose your sex drive and gain 40 pounds over here, but you might not be suicidal, and it might work for most people. Over here, they're more selective, but they're less likely to work for everyone, and they also have fewer side effects. Does that make sense? Okay, so serotonin receptors are called serotoninergic or 5-HT-ergic. This is also called 5-HT, 5-hydroxytryptophan. Serotoninergic. 5-HT-ergic. And you got a bunch of those different kinds of receptors because there's so many different places in the body that use them. So let's talk about, so we've got your SSRIs right here that we have. There's a whole bunch of them. There's probably more than this by now because it's only up to 2005. So what other kinds of things use the serotonin system? If your serotonin is low, it can manifest in depression or anxiety. It's slightly more likely to manifest, is this right? In the two sexes, it manifests more frequently as, I think, depression in males and anxiety in females. But that's right, isn't it? But there's no guarantee. And you could get both. They're not mutually exclusive. You're like, I'm totally in bed, but stressed out about it. But the other thing is that since this is used in the brainstem and it's used in sensory and motor function, also every single hallucinogen that we have ever found except for one works on serotonin. So LSD, psilocybin, magic mushrooms, all of those, what those do is they have a massive serotonin release all at once. And what happens is you feel amazing, but you also see things and potentially hear things, right? And all of these functions are sort of activated simultaneously with very, very small quantities of these drugs. So, and then ecstasy or MDMA, what MDMA does is causes a massive release of serotonin and also inhibits reuptake of serotonin simultaneously. So you feel really, really good, right, for a really long period of time. It also seems to cause a release of oxytocin, and oxytocin is your bonding chemical that increases trust in the situation. So one of the things that they have figured out is that MDMA, or ecstasy, which, by the way, originally, when it was originally created back in the late 70s or early 80s, before it was used as a club drug. They used it as a therapy drug. They used it as a therapy drug, for instance, in marital therapy. And when they came to testify before the DEA, one of the things that they said is the therapists were saying, one guided treatment of a couple with MDMA is worth a year of therapy. A year. The results were maintained for long periods of time. And so it got Schedule 1. And so in the United States, if something is Schedule 1, you can do almost zero experimentation on it. So you can't really gather data about it any longer. So it was Schedule 1 after that.
Why was that again?
It was actually really politics mostly.
It doesn't affect dopamine at all?
No, it doesn't tap into your dopamine response system at all. And there wasn't really any good data. There's a whole series on it I'll tell you about. So anyway, it got scheduled as Schedule 1. And then, of course, it was used as a club drug. They didn't want people using it as a club drug, and they were pissed off that that was happening. Got Scheduled 1 by the FDA, and then, of course, there was no approved medical use for it. And then, about 5 years ago, they started accepting minimal trials with MDMA for patients with PTSD, post-traumatic stress disorder, because one of the impediments to getting over post-traumatic stress disorder is if you can't actually get through the experience well enough to actually process the experience. And with the increase of oxytocin in that situation and the good feelings that come with MDMA, people were actually able to get through it. So first it was used on people who were survivors of violent crimes. And then the next round, they actually used it on a few soldiers. And now we are in phase 3. And the FDA actually said that it was a breakthrough medication for PTSD. Yeah, a breakthrough medication for PTSD. So what they did, and by the way, there's some interesting clinical trials with the last minute here. One of the interesting clinical trials, I read it when it was in the first stage of trials. So what they did is they had a control group and an experimental group that had treatment-resistant PTSD. And what they did is over, I think it was a 4-month period, but I may have the months wrong. They did therapy with a nurse and a therapist, and there was a nurse there because they were using, it was a double-blind trial. So once a month, they were using a medication. There was a nurse there, and then there was the therapist there, right? But they were doing therapy every week for the people, and then once a month they would use MDMA or placebo. Got it? Okay. So what they did is they did this for a period of 4 months, and then they continued the therapy without the MDMA after the period of 4 months. And what they found was a 65% improvement in the symptoms associated with PTSD. It's really hard to get anybody over PTSD. And the important part is that the results were actually consistent a year later and then 2 years later. So they actually kept the responses. And now, like, there's a clinical trial going on that was written up in Forbes and in the Washington Post about this soldier who had tried to commit suicide multiple times, and he's now in this PTSD study. It's the first time he's ever actually felt better. So it'll be interesting to see what that. Now, it's not going to be legal in Utah, until 50 years after it is. So if you're really jacked up, you got to move, man.
I don't know. There's a lot of people here who use drugs.
Yeah, but
Prescription drugs.
legal?
If they're made legal.
If they're made legal? So you guys can go read about it. Just look up the FDA and MDMA, and you'll find a whole bunch of information about it. There may even be clinical trials going on in Utah now. I don't even know. Maybe.
It's the opiates that are over here.
Oh, the opiates.
Yeah, those are the safe ones. Yeah.
So